And so if we understand that microbes influence our health profoundly, the question we can ask is, can we then manipulate these microbes in order to improve our health? If they have so much power over our biology, can we take some of that power back through understanding how they work and manipulating them in a way that benefits our health? And so one question that our lab at Stanford is very interested in answering is a very basic question. What is a healthy microbiome? What does a healthy microbiome look like? Because you can't solve problems that are rooted in a diseased microbiome if you don't understand what a healthy microbiome is supposed to look like. And unfortunately, for the past 20 years or so, much of the answer to this question has been answered using, in our opinion, a flawed model. And that model is, I think, perfectly illustrated by this Banksy art that he did. And the flawed model is that in the industrialized world, we have a human genome that is actually very similar to that of our Paleolithic or Neolithic ancestors. Our genome evolves quite slowly. You know, our generation time is about 20 years or so. The microbiome, on the other hand, adapts quite rapidly. Their generation time is about 20 minutes. So if you change what you're eating today, your microbiome will look different tomorrow. And that, from an evolutionary standpoint, was a very adaptable part of our biology. Here was an aspect of our biology that could really change rapidly depending on the food resources that we had at the time. Unfortunately, in the past, say 50 to 100 years, our diet in the industrialized world has shifted towards that which is basically unprecedented in the history of humanity. And this is a diet that's very rich in processed, sanitized, calorie-dense, but nutrient-rich food. And so by studying the microbiome of Western individuals, these are people that are consuming this kind of diet, we worried that we weren't really going to adequately answer the question of what makes a healthy microbiome. Instead, what we were studying is, what does the microbiome look like in people that are healthy right now, but we know are predisposed to Western disease? And so instead we wondered, could we find people around the world that hadn't made this shift in their diet yet? We're still consuming a diet similar to that of our pre-agrarian ancestors. And there are these people that exist around the world, and one such group of people are the Hadza of Tanzania. This is a group of hunter-gatherers that have a diet that's very similar to that of our Paleolithic and Neolithic ancestors. They don't farm, they don't store any food. So every morning they wake up and they go out and forage and hunt for what they're going to eat that day. And here's a group of Hadza that have foraged a bunch of tubers. So this isn't just a pile of sticks they're burning. There's actually tubers on here. So this is sort of the ancestral potato. And they're cooking these tubers, and this is actually their staple food. This is what they can rely on pretty much year round and what makes up the majority of their calories. And the tubers are foraged by mostly the women and children within the Hadza. And the men every day go out and they are the ones that are trying to get meat. So they hunt with bows and arrows that they make. Some days the hunts are successful and they will have a great meal of meat, but many days the hunt is not so successful and they subsist on basically a vegetarian diet. And so we wanted to understand what the microbiome of people like the Hadza look like. And so in this graph here, what I'm showing on the y-axis is the microbiome from people all around the globe, from Africa, South America, North America, Europe. And on the x-axis, what this graph shows is how similar those people's microbiota are to one another. So the closer the bars are to one another on this axis, the more similar their microbiota is. And what we found was traditional populations, regardless of whether those populations live in Africa or live in South America or Papua New Guinea, they all have a microbiota that's actually quite similar to one another and very distinct from that of people living an industrial lifestyle. So these are people in North America, Europe. Our microbiota all looks similar to one another and very distinct from that of these traditional populations. Okay, so what are the differences? Why are their microbiomes so different from ours? One, there are many things that differ. One of the most striking things is their microbiota is actually much more diverse than what we see in industrialized population. So they have many more types of species of bacteria living within their gut. So if you think of it like an ecosystem, theirs is like a lush rainforest. Ours is not so much. They also harbor species in their guts that we just don't ever see in Western populations. So the Western microbiota does appear to be deteriorated relative to the traditional population one. If I had to pick two images to describe how I see these two different microbiomes, the traditional microbiome would be this image here on the left, high diversity, high functioning, metabolic capacity of this microbiota, and their microbiota is very much tailored to the degradation of complex carbohydrates, likely from all the tubers and plant material that they eat. The Western or industrialized microbiota looks very deteriorated relatively. We're missing species, our diversity is low, and the capacity of our microbiota to degrade complex carbohydrate is also depleted relative to these traditional populations. So we've come up with this model for what we think has happened to the microbiota over our evolutionary past. Before 10,000 years ago, all humans obtained food through hunting and gathering. Agriculture is about 10, 20,000 years old. When agriculture was invented and humans shifted towards farming for most of their food, the microbiota had to adapt to that difference in food. And then about 400 years ago was when we started seeing the beginnings of industrialized food. And then just about 50 or 100 years ago was when processed and sanitized food became widely available in the industrialized world. And we think with each change in the way that humans acquired food, that the microbiota had to adapt, and that these adaptations has led to the deteriorated state of the microbiota that all of us in the West appear to be harboring. We actually did a mouse experiment to try to address this model specifically. And what we did was we took mice that harbor a human microbiome, and we put them essentially on a processed, sanitized food diet. And what we found was with each generation of these mice, their microbiota became more deteriorated. They lost more species. And after 4 generations, these mice had actually lost almost 2/3 of the original diversity that the first generation of mice had. So this was a mouse experiment that indicated that diet alone is a very powerful lever in controlling the diversity of microbes that we have in our gut. And when we remove complex carbohydrates, we can really deteriorate the community that we have. Okay, so I've painted a pretty bleak picture here. This is the point in the talk that I'm sure a lot of people feel desperate, like there's just no hope. But I'm hoping that moving forward now, I can show you some data that provides a more hopeful view. We know that the microbiota in the West is deteriorated. We know that this deteriorated microbiota is likely causing or contributing to many of the diseases that are becoming so prevalent in the industrialized world. And there are probably many reasons why this has happened. I've talked about diet, but we know sanitation influences the microbiota, medications, formula feeding, all these things. So the question is, how can we get from this deteriorated microbiota that's contributing to disease towards a microbiota that's more healthful and beneficial? Is that just through changing diet? Are we going to have to reintroduce some of the lost microbes that these traditional populations have that we don't have? And so my husband and I have started this Center for Human Microbiome Studies, where we're trying to develop humans as a model for understanding the microbiome. Our lab has done a lot of mouse experiments, and those experiments are great in that we can really dissect what's happening to the microbiota and the host in a mouse model. But at the end of the day, they are mice. And what we really want to know is how we can influence in a positive manner the microbiome for humans.